So we're going to keep moving through our pipelines. At this point, we have high-quality read data that's been aligned. We've gone through a lot of the pre-processing steps like mark duplicates and base recalibrator, base quality recalibrator. And so the next step is to be able to say the true variation that your sample has compared to a reference or to other samples. So as you probably are aware, for humans, we share most of our genome with other humans. And in fact, most of the sites that are variant in any individual sample are common variation that are biallelic. So the key feature of these variations is that you want to be able to compare them across many different samples. And so for that, we're going to be using a reference. All of the variant calls that we make will be with respect to a given reference. It's really important that whatever reference you choose, you stick with that for your entire analysis. So there have been many reference builds for humans over the years, starting back in 2003. And then there's been iterative refinement over the years of these reference builds. So the latest and greatest is GRCH38. And that is what we would recommend using today. And with each reference build, you've gotten higher quality alignments. They've fixed mapping issues. And in GRCH38, we also have alternate contigs, which is kind of like the latest and greatest for this particular reference build. And those alternate contigs allow you to be able to capture the highly variable regions of the human genome. So let's walk through some examples. This is a cartoon of what all of the different kinds of variations that we're going to want to be able to detect from our read data look like. So we have, let's start with short variants. Here in this cartoon, we have the reference sequence at the top. So here we have chromosome 1 in orange, and this is our read pileup. And we can see at this one particular site, instead of an A, we now have a C in our sample. So we would think of that as a point mutation, a SNP. And we could also have indels, which are small, less than 50 base pairs typically. Something that is either a small insertion or a small deletion that you could detect from short read data. So this very small deletion is missing a piece of the DNA that was in the reference, but it's small enough that we can see it within a single read. And that's kind of like the barrier of where we would consider it an indel versus a larger event, which would be a structural variant. Structural variants can include copy number variation. And when we talk about the pipelines that we use to detect copy number variants, we're looking at read depth as the signal that we want to make that call. So for example, in this sample, this piece of the reference doesn't exist in this particular sample. And so because of that, we don't see any read coverage over that region at all. And you can also have a hemizygous deletion where we're seeing half the coverage we would expect. And you can have a gain, a copy number alteration, where you see more additional copies of that event or of that region in your sample. You can also have a larger subset of variants, structural variants, that can be really massive. And you could have things like translocations where a piece of chromosome 5 in this example has attached itself to chromosome 1. And you can see that this spans across either a read pair. So you'll see that there's one read in a pair is aligned to chromosome 1, but the mate is aligned to chromosome 5. Or in some cases, you'll see split reads. There's different kinds of evidence that we're going to have to put together and piece together to make these calls for these large events. In addition, we also have a tool called PathSeq that can detect pathogenic sequences. And this is your unmapped reads, the things that are not human, but you might want to know what kind of bacterial contamination, for example, you have in your sample. So that's another possible path forward. And all 4 of these different kinds of variants come in 2 different flavors. Germline, which means that you were born with that. Your DNA does not change over the course of your life. That would be a germline variant. It's still different from the reference, but it's not changing for you. And then somatic variations, which you accumulate over time. And it turns out that the evidence that we're going to need for these 2 different kinds, germline and somatic, are going to look very different. So you're going to use an entirely different pipeline, entirely different set of tools to call those variants. And please stop me and ask questions in the middle of this talk if you'd like. Sorry, should have mentioned that earlier. So here, if we look at the exact same kinds of variants we just saw on the previous slide, but now we're looking at an IGV screenshot. Here, the reference is now at the bottom. This is the reference genome, and if it's gray, I think Mark mentioned this earlier, but if these reads are all gray, it means they match the reference. And when we see color, that means that it's not matching the reference. At the very top, we see a coverage track. So this is just counting the number of reads that all align at this site, and that's what you're seeing here, is that histogram of that. So if we start over on this side, we see a probable insertion. There's an eye drawn on that IGV puts here, and if you were to click on that eye, it would expand, and you could see the bases that had been inserted in that read. And so that's probably an insertion. We see that at this pileup, we see a lot of these eyes. We also see a probable SNP. Here, there's kind of an orange color and a blue color, so this is probably a GC het site. And you can see they're all just piled up right there. And you can see it in the coverage track as well. It will highlight that in a different color. And here we see a probable A SNP, where it's all green in this site. But here, where everything is lit up and colorful, we're seeing a lot of bases that don't match the reference. Most of these reads have been soft clipped so that half of the read doesn't really align to this location, but the aligner caught that the other half that's gray does belong here. The reason that we think this is a structural variant and not just the sequencer fell over and kind of gave up at this spot is that they match each other. So the reads, you can see that this red section kind of traces down for all of these different reads. So that indicates that this is something real. The DNA was read properly by the sequencer, but it's actually belongs somewhere else in the genome. You can also see that the coverage drops in this region a little bit. We're going to see that read coverage is a pretty messy signal, and so we're going to do a lot to clean that up, but this could be a deletion as well at this site. All of the variants will be reported in the VCF format, the variant call format, which the basic format looks like there's a header with lots of metadata information about the samples that you have in there, and information about the annotations that you've made about your sites. And then you have just a tab-delimited format where the first 9 columns represent each site. So you have the chromosome position, et cetera. And we have different annotations. So for example, here DP means depth. So at this site, you had 14 reads to make this call. Following the format field, you have a different column for each of your samples. So this is a multi-sample VCF. It has 3 different samples, 1, 2, and 3. And here you can see the genotype for each of these samples. So at this site, in chromosome 20, the first sample actually does not have a variant there. It's HOMREF. The genotype is HOMREF. But you can also see that the second sample does. It's a het site at that location, and then the third sample is HOMVAR. It has 2 copies of that allele. Yes?
What's the difference between, I think you said read depth versus different, those columns after it? NA, 01, 02, 03?
What's the difference between depth?
What are those ones called? The 01 versus 02?
So each of these are different samples. So NA, yeah, sorry. So before this column, so before the format column, everything is about the site level. So now you're talking about a specific place in the genome across all of your samples. But once you get to these columns, now you're talking specifically about sample number NA0001. Yeah, okay.
But which samples depth is reported?
I think that's global across all of the samples. This is a cartoon example, so typically you would have higher depth than 14. If you had annotations specifically about an individual sample, it would be incorporated in this place here, and the format field is telling you what these numbers mean. So GT stands for genotype, and it's telling you look at the first number in this colon-delimited map. We can also store structural variants and copy number variants inside of a VCF. It gets a lot more complicated, but you can see here that the alternate allele is a symbolic allele. It has, you know that because it's in this bracket. So when you see that, it means that there's something complicated going on here. You're going to get a lot more information in the annotations that you've stored about that site. But we are able to store these calls and the breakpoints in the VCF. So we'll quickly go over the high-level ideas of all of these different pipelines. There's different pipelines for every kind of variant that we've talked about. So for example, for germline SNPs and indels, we're going to use the haplotype caller pipeline. For somatic SNPs and indels, we'll use MUTEC2. They're based on some similar code. They share some concepts, but they are going to treat the data very differently. And we'll see that similarly for copy number variation and for structural variation.
Sorry, so
Yep.
those are two different tools for copy number variation?
For copy number variation, there are two different pipelines. There is a germline copy number variation pipeline, that's the GCNV, and then there is the flagship GATK CNV, which is called somatic events, as well as allelic CNV is what the ACNV calls. This is the germline SNPs and indels pipeline. I think we're going to be talking about this on day 2 in more detail. But the basic idea is that a single genome, when you're studying germline calling, it hardly ever will help you if it's in isolation. You always want to be comparing that sample against other samples, either in a population or possibly in that sample's family. So for example, if you are looking for a rare disease, you want to make sure that it's a rare variant. And you possibly want to know that it's de novo, that the parents of that sample do not have that mutation. So there's a couple ways you could do this. You could make individual call sets and then compare them afterwards. But we would actually recommend calling all of your samples together because this gives you more power. It makes your variant calls higher quality if you can look across multiple samples at once. So the pipeline that we're going to describe is going to use a joint calling idea. So the first step of the pipeline, yes?
Just a quick question.
Yeah.
Is there some empirical measure on how many samples would you require to jointly call in order to start getting benefits? Like, clearly, the more the better, but
Yeah.
Does it matter if I have like 100 samples or 1,000 samples? Is there a huge...
So in the germline context, I think that if you, I think in the hundreds, is that right? I'm looking at Geraldine, sorry.
We'll talk about that in more detail tomorrow.
Okay, but even 100 samples is still well worth it to do a joint call set, yeah.
Why do you need to do a joint if you have a reference genome? Isn't that the point of the reference?
So the joint calling, the question is why do we need to do joint calling if we're still comparing to a reference? The joint calling is going to use the actual read data from many samples together to give you more statistical power in calling the variants. The reference is just a static string. It never changes. It's just for a given reference build, it just says that at this site there's an A. And then for each of your individual samples, you actually have read data that's going to tell you, oh, actually, it's pretty common that there's a G here in the population, so that's going to inform the variant calls we make at that site. Does that? So the first step of this pipeline is to run individually on each of your samples, to run haplotype caller to get a GVCF, which is a genomic VCF. And the reason it's genomic is because it has information at every single site in the genome for that sample. And so here in blue, we're seeing these reference blocks, which tells us how confident we are that the sample was truly referenced at that site. It could be that you just don't have much coverage for whatever reason over a certain block of the genome. So there you wouldn't want to say you're highly confident that your sample is referenced. You would want to say it's a no call or something else. So that's the information we don't want to lose, even though we're running individually on each sample at this stage in the pipeline. Then we take all of these GVCFs and we run a joint calling pipeline with genotype GVCFs to actually genotype each sample. And then we have removed the reference blocks, so we no longer have information across the entire genome. We now just have the sites where at least one of your samples in your joint call set was variant. So this makes your file size smaller. You don't want to have to cart around lots and lots of genomic region that matches for every single person to the reference. Once you've run the joint calling step, then you're going to want to do variant filtration. We've tuned haplotype caller to be as sensitive as possible, so we didn't want to lose any true positive sites that might be out there, but that means that we let in some false positives. We want to be able to use the annotations that were stored in that VCF to filter out some of the possible false positives. And this lets you tune how sensitive you want to be for your individual analysis, because it can be different for every person. So that's why we're making it available that the variant filtration happens after being as sensitive as we possibly can. So here in this plot, we're seeing two examples of annotations on the X and Y axis. And you can see that if we were just to hard filter them, if we were just to cut and say everything to the left here is a false positive, so we're just going to filter those out. That's not giving you a fine, you're not being able to separate as clearly if you instead used some kind of machine learning algorithm that could look at many of these annotations in higher dimensions than just 2 in order to color the variance, which is what we're seeing here. So here we're using VQSR, the variant quality recalibrator, which is saying that all of the red dots here are really high quality, and then we get lower and lower quality as we move away from that cluster. And we're also going to learn more about some deep neural nets that are also in the mix for making our variant filtration step in the coming days. Yes?
Just this looks to be done now. Are you planning to develop a more visualization tool into GATK4 or any plan to have more visualization? Because I think it's a little bit messy.
Yeah, it gets messy. So I don't know of any specific plans of focusing on making visualization tools in GATK. I know that our copy number pipelines do output some plots. And then once you have your VCF, you should have the annotations and everything you need in that file format to be able to make your own annotations. And I think we'll see that in some of the hands-on tutorials throughout the rest of the talks. And you'll be able to use notebooks in Terra to be able to make these R plots that are really beautiful. And I think we'll see that too. So finally, on the Germline side, one more pipeline you might want to do. This is a pipeline to get you the highest quality variant calls that you can get. This is to narrow down to find possible de novo mutations that might be causing a rare disease. So here we have a pipeline that calculates genotype posteriors by using information from a pedigree file from your family of the sample, as well as population information. And we can filter it down to get just the highest quality variants in order to get just, in this particular case, 8 high confidence de novo calls, which makes it a lot more feasible to follow up on those 8 calls and see if there's something valuable to find there. Finally, we have a new tool called Funcatator, which adds functional annotations to your VCF. This allows you to know what kind of change in the biology is actually happening because of this variant. So it'll annotate each of the variant sites with things like what gene it's from, if it's a synonymous mutation, if it will actually make a difference to the protein. So here we see that the majority of SNP calls are actually in the intron. But you can see that as you narrow down, you can find all of these different annotations to let you focus on what really matters the most for your sample. So that's the Germline side. So we're going to move on to Somatic unless there's a question. Okay. So for the Somatic pipeline, the situation is a little bit different. Here in this cartoon, we can imagine that we have some normal cells, we have dysplastic cells, and some cancer cells. We have purple and blue. There's 2 different subpopulations of cancer in this tumor. So when you take a tumor sample, you end up with a mix of all of these different cells all together. So this causes problems for analysis. You wouldn't be able to use a germline caller because now we expect contamination from the normal. So we're no longer expecting a ploidy of 2, a beautiful 0.5 allele fraction for your call. It could be much lower because you have the normal sample mixed in. In addition, your cancer cells could have multiple populations. And so then you'd again expect low allele fractions to come into play because you have many different populations all mixed together in the bulk sample that you've sequenced. So our expectations in the germline side were that we have this beautiful signal and not very much noise. Now we have heterogeneity of the tumor sample. There's multiple subpopulations. We have contamination. We still have the sequencing noise, and our signal is a little bit lower. So the way that we're going to try and combat these issues is by using carefully selected controls, normal samples, that we can remove some of the artifacts that would have been called if we had not had them. So if it's possible, the best thing you can do is run this pipeline with a matched normal. So you have a tumor sample and you have maybe blood from that same person. You could also use a panel of normals. You can use both of these together. A panel of normals is a bunch of normal samples that you've run on a very similar technology to the tumor sample that you have. And that will help you remove some systematic errors that you would see across all of the samples, either due to sequencing or possibly mapping artifacts, things like that. So here's an example where you might have a matched normal. And here at this site, we see a bunch of colors, so that means that it's not matching the reference. This is possibly a somatic SNV, because there's nothing here in the match normal. Whereas at these other sites, there's something going on in the match normal, so we would probably want to filter those out. But we also could use a panel of normals. Here you can see the tumor has some kind of artifact. There's this blue here, and it's kind of smeared across. It's probably a sequencing error. But we don't see anything in the normal. Maybe the normal had a better luck with the sequencer on that day. But hopefully, if we have a panel of normals that's used that same sequencing technology, some of those would still have a very similar looking artifact. So we'd be able to filter this out. Similarly, you could have a germline event, so that should be present in the normal. But for whatever reason, the coverage of your normal is low at this site. You don't have much here. So if it's a common variant, which most of your samples variants will be common, maybe you can catch that by seeing it in a panel of normals. So this is what the pipeline currently looks like. You can have a tumor normal pair where you run MUTEC2. And then similarly to how the germline pipeline worked, you're going to be able to filter the variants after running MUTEC2. MUTEC2 is designed to be as sensitive as possible. Then you can filter to however much you need to do. Somatic, yes. Oh, nope, OK. Any other questions? Yes. If you don't have any normals, you can still run the pipelines, but you might get lots of false positives. Yes, yeah. You can run this pipeline without a matched normal, and then we would really recommend using a panel of normals if possible. Similarly, if you only have a matched normal, you don't have a panel, you can do anything you want to do. But the more normals you have, the higher quality your calls will be. Yeah?
Do you guys have a panel of normals that would sort of be a standard set of panel of normals that people frequently use?
Yes, I believe that if you are getting your sequencing done at the BroadE, we would be able to provide that. Is that, stop me if I'm wrong on that. I think we have that. Yes. If you're doing sequencing somewhere else, then you should really try and use as close to that technology as possible. You could still use a panel of normals provided by the BroadE, but it might not be as effective because the artifacts will look different. Yep.
I think it's important to comment that the panel of normals isn't just about the population, it's also about the technology that you're running as well. So the normals should be run with the same sequencing machine, the same sequencing chemistry.
Yep. Just to repeat, the technology that you're sequencing with is really important. You're trying to filter out sequencing artifacts. So in order to do that, you need a panel of normals that is run on the same technology. If you were just trying to filter out common variants, you could use something like Nomad. And we would recommend doing that, actually, especially if you don't have access to a panel of normals or a matched normal. But the panel of normals is really helpful for removing sequencing. Yep.
So it sounds like so I'm actually interested in germline copy number variants. And it sounds like this is less of an issue for the germline, right? You're talking specifically about single nucleotide.
Yes. Right now we're talking just about single nucleotide polymorphisms. But if you're going to hear the words panel of normals used a lot, it actually means a very different thing for the MUTEC pipeline than it means for the copy number variation pipeline. They're both using the same concept that we want to remove artifacts that we see in a bunch of normal samples. But they're still different. You would need to create them differently and use them differently. Yeah?
Yeah, there's an idea that's floating, and we'll see if we can push it through, but to kind of, fair point.
There's a potential for us to develop something that will be a more universal panel of normals for this application. And at that point, we would call it something else because the panel of normals confusion has been hitting us a number of times because they're so different between different applications. And the proposal is to call this new thing the common recurring artifacts panel. Check out what that spells. I like it. Anyway, we'll filter out that.
Great. Awesome. OK, so moving on to somatic copy number variants, where we're also going to hear about panels of normals. So copy number variants, as we saw before, we can imagine where you would expect your sample to be diploid. You would have 2 copies of any given gene. So in your normal tissue, for some control gene, you have 2 copies. And for TP53, you also have 2 copies. But in your tumor sample, you might still just have the 2 copies of your control gene. But TP53 now has 5 copies at this region. And that's what we're trying to detect. with this pipeline. And the evidence that we're going to use to detect these variants is coverage. So it's just counting how many reads were at a site. That's the evidence that we have to work with to make these calls. So here in this plot, we see the x-axis here is genomic region, genomic location, and here we have total copy ratio. So for most of this sample, we see the copy ratio of 1, so that's just, we've divided by 2 if this was a human sample. But we also see this jump up here, that's probably a copy number event where there's been a duplication of that region. And here, similarly, we see a deletion. Now, if you take that same plot and rotate it, so now the y-axis is genomic location. This over here is the raw coverage. This is the kind of data that we're starting with. It's clearly very, very messy. I don't think I could look at this and say, oh, for sure, that looks like a copy number variation. So the bulk of this pipeline and how it's really useful is in standardizing and denoising and making the data clean enough to be able to make a confident call based on the coverage. So in the standardization and denoising step, that's where you would use a panel of normals in order to remove all of this messiness and make it look more like the final output here. Once you've denoised, then there's a segmentation step, which will break it up into chunks so that you can say, this chunk is where I'm going to call an event. So here we see maybe there's some called events in this region. And finally, we can also look at heterozygous SNPs across the same regions. And if we were to see some kind of copy number event, we would expect that the minor allele fraction to not be 0.5. We would expect it to be lower because there's been some kind of alteration there. So that's just another way of looking at the data in order to be able to see that this is something that's happening in this region. So we'll talk more about this pipeline on day 3, I believe. So the newest methods in the GATK include a germline copy number variant caller. Calling germline copy number variants is actually even more difficult than calling in somatic. That's because they are rare. In somatic, it can be quite dramatic. The copy number events can be very long. There can be many of them. In addition, it's hard to know the difference between a common copy number event and something that's a sequencing artifact. So it's difficult to tell when you're trying to call common germline copy number events. So this pipeline runs differently from the somatic version. It does the denoising, segmentation, and calling all simultaneously. And they've had some really good results, so I'd highly recommend checking out this pipeline and using it. Finally, possibly the most difficult kind of variant to call are structural variants. Here in this cartoon example, we have a reference where there might be these 4 different pieces of DNA. And we can have all different kinds of structural variants that arise at this location. So similar to what we've seen with the copy number variants, you could have duplications where you have additional copies of B, but you might want to know where they ended up. So you might have a tandem duplication where they're all next to each other. Maybe one of them was inverted. It could be dispersed where one ended up in an entirely different location. You could also have a deletion where that B region was missing from your sample. But you could also have structural variants that don't involve copy number variation. So here we have an inversion. There's still only one copy of B, but it's moved over and now it's inverted. We could have translocations where something from a very distant location, like chromosome 5, ended up in chromosome 1. And structural variants are especially difficult because in real life, you can have almost any combination of all of these all at once. And so it gets very complex very quickly. So the pipelines that we're developing for structural variants are trying to find the breakpoints of where these events happen and piece it all back together. So that's most of the pipelines for all of these different kinds of variants. And we're going to go into a lot more detail in the coming days of all of these different pipelines. You can also find all of the pipelines fully coded out in this GATK workflows repo. They're all hosted here as WDLs, which we're going to hear more about in the coming days as well. But it should be very easy to take them and then use Terra in order to take your sample and just run this pipeline on your sample. So you don't have to reinvent the wheel, which is great. You can just tweak the inputs that you have with your sample and then run the pipelines super easily. So we're going to see examples of that in the coming days. And that's all I have. So if there's questions, yes.
So in pediatric oncology, a lot of times we'll sequence the tumor and we find mutations and we want to know whether they're in the germline. So we also have the germline, and it's been done clinically. But the variants have been called, obviously, with the germline pipeline rather than with the somatic pipeline. And how confident are we that if we're using two different pipelines, because that's what's clinically accepted,
Right.
the somatic versus the germline, that the germline is going to find something that we found in the somatic caller if it isn't the germline?
If it's truly a germline event, then I would use haplotype caller and be very confident that that is a true germline call. You would still want to use that matched normal when running your somatic pipeline in order to filter out as many artifacts as possible, just from the get-go. So I would still recommend using it with your somatic pipeline if it's possible. But then if you wanted to do a further analysis after the fact and look at the germline calls, you could absolutely use haplotype caller to do that. Yeah.
Could you comment generally on how you assess like the quality of any of your calls, like not the nitty-gritty, but just like what are the guiding principles? It's like a lot of these tools are, you know, getting really complicated, and I do think that it's important to understand like how it's working, but it's also like just how you assess quality.
Yes. So the question was, how do we assess quality of our final VCF outputs? And we're going to go into a lot of detail on that for germline tomorrow, and then for somatic on day 3. So I don't want to get into the nitty-gritty, but there are definitely ways across your whole sample. There's QC metrics. Across a cohort, there's QC metrics. And then we're also going to look at how you can make plots from your VCF of those annotations in Terra or something like that. And so we'll definitely get into that. It's a big question. Yeah.
To also piggyback on the question on pediatric cancer, so what you do just MUTEC2 for somatic variant calling, you also get like, you know, so you get those variants that pass filter that I guess you're pretty confident are somatic, but then you get those that I guess are a variant, but they didn't pass the filter.
Yep.
And so, you know, I've been kind of treating them essentially as germline variants without ever running the haplotype caller.
Yep.
Is that fair?
Yes,
to do that,
definitely
or?
fair. So the question was how to understand the filters that are coming out of the MUTEC2 pipeline. And it should have filters that will say germline event. So if you look at the filters for each of the sites, you should know why it was filtered out. So some sites will be filtered out because there wasn't enough evidence. Some sites will be filtered out because it's likely it's a sequencing artifact. And it will tell you that in the filter. Or if it's a germline event from your match normal, it will tell you that there too. Yes?
So I guess all these tools have been tested on whole exome or whole genome.
Yes.
You apply this on, for example, targeted sequencing only covers, for example, hundreds of genes with higher coverage, but just fewer regions.
Yeah. So I think you can definitely still run these pipelines. It might take some tweaking of some of the parameters, and maybe we can talk offline about the specifics of what your data looks like.